Imagine a cancer that's, well, incredibly aggressive, often found too late, but at the same time, it's really stealthy. It can actually hide from your body's own immune system.
Yeah, that's Cholangiocarcinoma, or CCA. It's a malignancy that forms in the biliary tree.
That network of ducts carrying bile from the liver.
Exactly. And it's unfortunately one of the deadliest cancers. The 5-year survival rate is grim, hovering around 10%.
Wow. And for advanced cases?
Median survival is maybe 12 to 15 months. It's tough. Okay, let's dig into this.
And a big part of why it's so tough is this immune evasion you mentioned.
Absolutely. It's not just passive hiding. CCA actively manipulates the immune system so it can survive and, spread without being challenged like it should be.
Okay, so our mission for this deep dive is to really unpack how it does that. We're looking at some new research that sheds light on the specific ways it evades immunity. That offers real hope, right? For new diagnostics, new treatments for you.
That's the goal. Understanding the mechanism is key.
So this immune evasion, it's how the cancer cells really thrive.
Yeah.
But you said it's not just passive hiding, it's more active.
Precisely. The cancer cells actively create an immune-resistant microenvironment. Think of it like they're building a shield around themselves.
A shield. Or maybe even recruiting guards. How do they do that? What are they using?
They use what we call bioactive molecules. These are things like cytokines and chemokines, tiny chemical messengers.
Okay.
And what's really fascinating, maybe a bit scary, is how these molecules hijack the body's own immune cells. Specifically, they reprogram macrophages.
Those are the immune system's cleanup crew.
Right, yeah. They reprogram them towards what's called an M2 subtype.
And M2. That's bad in this context.
Very bad. M2 macrophages are immune-suppressive. They actually help the tumor. They have a pro-tumor phenotype.
So the body's defenders become collaborators.
Yeah.
Helping the cancer grow and hide.
Exactly that. Instead of fighting the intruder, they're actively helping it set up shop and expand. It's quite a strategy.
Chilling, really. So the research we're looking at tried to figure out how CCA sends these reprogramming signals.
Right. The study aimed to understand these immunosuppressive characteristics by basically listening in on the chemical messages. They looked at the stuff released by different CCA cell lines in the lab, the culture supernatants.
From different types of CCA,
right,
like intra and extrahepatic.
Yes, exactly. And they checked how these different chemical cocktails affected the M2 programming of fresh human immune cells, monocytes.
And what did they find? Were the messages the same?
That's the crucial part. No, they weren't. Each CCA cell line had its own unique profile, its own signature of cytokines and chemokines.
Okay.
And interestingly, only the signals from 2 specific cell lines, RMCCA1 and HusCCT1, were actually able to induce that M2 polarization.
Only 2 of them? Wow. So not all CCA uses the exact same trick.
It seems not, and preliminary work suggests it might even be specific fractions within those signals doing the heavy lifting.
So different types of this cancer might have, unique molecular fingerprints for how they manipulate our immune system.
That's what it points towards. Distinct molecular fingerprints. And that's huge.
Huge how? What does that mean for, fighting this?
Well, if we connect this to the bigger picture, understanding these specific mechanisms of immunosuppression is absolutely vital. It stops being this one big, unbeatable enemy.
Right.
It lays the groundwork for developing potentially novel diagnostic tools, maybe ones that can detect these specific signals early on.
And better treatments, too.
Exactly. More targeted therapies.
Yeah.
Instead of a broad attack, you could potentially design something that counters the specific immune evasion tactic used by that patient's type of CCA based on its fingerprint.
That sounds much smarter. More tailored, personalized approaches. It feels like this research really opens up some genuinely new and promising avenues.
It really does. Which leads to, I think, an important question for you to consider. If we get really good at identifying these unique chemical signals, the specific way each cancer recruits those M2 macrophages, could future treatments become less about, say, broad chemotherapy and more about subtly disarming just that one specific immune evasion pathway? Almost like cutting the cancer's communication lines, cell line by cell line.
Reprogramming the reprogrammers in a way.
Perhaps. The key takeaway is that by understanding the specific molecular conversation, we're finding the cancer's unique voice and figuring out how to potentially silence it.
It's incredible to think about the power in understanding these tiny, specific interactions. It really could shift how we approach fighting such complex diseases like CCA. Whole new angle.